Now I'm happy to introduce Dr. Pamela Marrone of Marrone Bio Innovations. Dr. Marrone spent her career focused on natural products for pest management for the past 30 years in Davis, California, where she started and led 3 biopesticide companies. She started Marrone Bio Innovations in 2006 to discover and develop bio-based products for pest management and plant health. On August 2nd, 2013, MBII was listed on the NASDAQ. Before Marrone Bio, she started and led AgriQuest. Before AgriQuest, she was founding president and business unit head of Intotech Incorporated. She started her career after graduate school at Monsanto, where she led the insect biology group. She's a trustee of Cornell University, treasurer of the Association for Women in Science. She mentors and advises a group of ag bio and ag startups. She has a BS in entomology with honors and distinction from Cornell and a PhD in entomology from North Carolina State University. And now we welcome Dr. Marrone to begin her presentation. Pam.
Thank you, Steve. Welcome everyone listening to this webinar today. I'm sure we have a higher attendance today because many of you are sheltered in place, so have at home and wondering what to do, and listen to what I'm going to say about natural products. This is the safe harbor slide. I just got to show you this because we're a public company. I started my love of natural products actually way back when I was little. I grew up in Southern Connecticut in a beautiful place in the woods, and my dad actually used the first ever biopesticide ever commercialized based on Bacillus thuringiensis, which is a microorganism of bacteria that disrupts the gut of caterpillars. And he used it against the gypsy moth that was destroying this dogwood tree, which was very little at the time and now is still there. And my mother still lives there at age 93. And this dogwood tree outside the kitchen window. So this Bacillus thuringiensis, a marvelous example of a biopesticide, still on the market today, and about $500 million of sales every year. Now, despite being in a COVID-19 world, it is a great time to be in agriculture. We are actually a business that is critical for food, obviously for food production in the world. So farmers are continuing to grow their crops, and all the associated businesses like ours are conducting business during this crazy COVID-19 world because people have to eat. But there's a whole explosion of new companies and startups and money, $8 billion worth, going into companies in this industry, from farm to fork, improved crops to crop inputs like our companies, biostimulants and biopesticides, and precision farming, big data, robots, and of course improved protein sources and burgers made from plant-based substances, as well as insects for protein. And so this is a great time to be an ag if you're coming out of school or you're a postdoc and you're looking to join companies. Please look at the startup world, because there's a number of well-financed startups that you can join, and definitely for chemists and biochemists and biologists and chemical engineers. There are so many pesticide issues today. I have listed all of the many ones, and the ones in red are the ones that are always top of mind. And while it's COVID-19 all the time in the press, there's still things that are published about the various pesticide issues that are going on with glyphosate or pollinators, neonicotinoids. California banned chlorpyrifos. I served on the chlorpyrifos alternatives task force recently, and so forth. So never a dull moment in the pesticide world. And it's harder and harder and harder to find a new chemical that can be commercialized that meets all of today's environmental and social economic requirements. It costs, as a result, then almost $300 million, and takes almost 12 years, 10 to 12 years to get a new chemical pesticide to market. And if you look at the bar graph on the right-hand side, the number of, the green is the number of launched products, and the brown is the number of new leads. So there's a lot of new leads coming out of the various company labs, but many fewer. There's a little uptick of late, but there's certainly not a huge pipeline of new active ingredients coming onto the market, leaving a good opportunity for biologicals. The big companies have merged to form the top ones, Syngenta, Bayer, BASF, Corteva, UPL, and FMC in that order. And so we've got some big companies now, but the chemical pesticide market is growing at single digits. That's because of the low commodity prices, global regulatory restrictions, pest resistance, and the high development cost, the barriers to entry. Meanwhile, the biopesticide, bioprotection crop input category is the fastest growing category, growing at double digits. While one would think that parallels the fast growth organic food market, which is the fastest growing category of food, actually most biologicals are used in conventional agriculture, and I'll explain that in a minute. Big companies have seen the opportunity in this area, although I think they probably think there's yet to be realized in the biological arena, despite purchasing a number of companies in the past decade. And here are some of the deals that have been done. The three categories we have of biologicals are here on this slide, but I will talk primarily about biopesticides. I don't have time to go into the other two categories of biostimulants and biofertilizers. But biopesticides, it's a term primarily used in the United States. Outside the United States, you'll hear biocontrol, but the latest word that the industry likes to use is bioprotection, just like the agrochemical industry uses crop protection instead of pesticides. Now, on the biostimulant side, we're talking about natural substances or microorganisms that enhance crop growth or reduce crop stress. This is also a double-digit category. However, it's in flux as far as the regulatory process, because there's a patchwork of regulations by state in the US and also around the globe. But there's some harmonization work going on. There was a definition of biostimulant in the 2018 Farm Bill, but EPA also has put out a Federal Register notice of their intention to regulate or clarify the regulation for biostimulants. So this is an area that's causing some confusion about whether a seaweed extract is a plant growth regulator or a biostimulant under the biostimulant rules versus being regulated by the EPA as a plant growth regulator. So stay tuned on this area. The biofertilizer area, it's called bionutrients now, is also a fast-growing area. There's a number of companies that are now looking at maybe gene editing microbes to produce nitrogen, or just taking naturally occurring microbes to create to have microbes that can provide nutrients or enhance nutrient uptake. So this is a very focused area for a lot of companies right now. The biostimulant area, because of the lower regulatory barriers, is very crowded, a lot of companies, and so it's important to look at companies that are very science-based in this arena and how they're distinguishing their science from others. As I mentioned, often biologicals are seen just for organic, and they're pigeonholed that way, but more than 70% of biologicals are used by conventional growers. So conventional growers are picking these up at a rapid pace, and why is that? Well, biologicals and chemicals together in integrated programs can offer better performance and return on investment to the grower than either alone. Our company has data over and over again to show that. And today's products are much higher performing and more science-based than they were. I'll have some university extension, cooperative extension personnel who tested my product 5 years ago say, oh, I tested that. It didn't work. And I said, well, you know, every year our products get better because they're often based on microbes, and we can continually tweak and improve them. Plus, we have all the tools today, modern tools of molecular biology, We can gene sequence the entire microbe and learn what it's what it's doing, what it's making, and its physiology. And there's a lot of new formulation technology that's being able to stabilize or encapsulate natural materials. So today's products are very different than they were even just 3 years ago, 5 years ago, or 10 years ago. So that's something to keep in mind. We're also finding that the grower awareness and knowledge of these products is increasing, although still rather low relative to chemical products. So there's still a lot of education on how these products work. They're new modes of action, and so therefore they're great for managing pest resistance. But they also you have to educate the grower and the user on how to use them, because they're often not like chemicals in terms of their mode of action. Not quick knockdown, they can be slower acting. So I'm going to give you some examples of that. Great advantage, you can spray right up to harvest and export and go, not worry about maximum residue levels because they're exempt from MRLs and tolerance exempt, and also exempt from global codex rules which govern the chemical residues globally. In this time where, especially out in California where I live, getting labor for farm labor is very difficult. And so when you spray in the morning and can be back in the field with your crew is a great advantage for these products because you have fast field reentry. And generally, these products are low risk to pollinators, people, and the environment. And with the ever increasing consumer demand on food companies for transparency and sustainability of their food supply and the food they eat, this is a benefit in helping farmers and food companies meet their sustainability requirements and metrics that are often being imposed upon farmers. We did a study last year with the help of the UC Davis Graduate School of Management that looked at our products based on microorganisms and plant extracts and compared that to our competitor chemical pesticides, and found that we could reduce our carbon footprint by about 60 to 90% with our biologicals. So this is a very good benefit, especially when, again, what consumers are looking for, but also investors are looking for companies to increase their ESG score, which is environmental, social, and governance. And there's a lot more scrutiny on this with public companies, all companies. And we can get a product to market for less than 10 million for the US launch, maybe another 5 to 10 million for a global development, and 3 to 5 years. So much less than a chemical pesticide. So speaking of that, it's really important to understand the differences in the business models of the two types of products. Chemical pesticide, 10 to 12 years, almost 300 million from discovery to launch. Massive upfront capital, thousands of global field trials. So you have your label, pesticide label built out, and then there's a global launch big, large marketing spend, and peak sales in a relatively quick time. This is very different than biopesticides, typically because biopesticides are the domain of smaller companies like ours. So we can get a product to market in a few years, and as I said, less than 10 million. But when we launch it, we'll have a much narrower label than you might for a chemical, because we have a shorter period of time where we've been able to generate that data. So we might have 150 or 200 field trials, not thousands, and we might have 10 to 20 crops on the label or pests versus hundreds that could be over time. It's capital efficient. We can pay as we go and sell, and then move, and then as we get a feedback from our early adopter growers, that feedback can go back into new versions of the product. So we'll launch with version 1.0. It's a good product. Growers want it, and there's a need in the market, but we know we can improve it. We can improve it in our own cost, in our own profit margin, but also cost effectiveness to the grower. We can come up with new formulations. We can reduce the use rate because we can enhance the microbe yield and potency, and therefore go from 4 quarts to an acre to 1 quart an acre, for example, and continuous improvement. So every year we can actually have some improvements in the product, but since big improvements require a new EPA registration. We might have a new product every 3 years, for example. And this is important for our customers to understand. And we get a lot of early adopter growers who love to get something early and help shape it as we improve it and add more crops and pests to the label as we go on. But it's important as practitioners to understand the difference in business models. So when a product biopesticide comes to market, they understand the stage at which it is in its development process. It's not going to be as massively developed as a chemical pesticide, but still good enough to serve a market need. So what are we talking about on the bioprotection or biopesticide biocontrol in terms of regulatory process? Well, there's two branches at the Biopesticide Pollution Prevention Division at the EPA. And there's microbial branch and there's the biochemical branch. Microbials, just what they say it is. Microorganisms used to control a pest or pathogen or weed. And they can also be dead. They can be alive or dead. And so this is a generally unique to the US, that dead microbes can be registered quickly, and often dead microbes outside the United States will have more requirements because they're seen more like chemicals. Biochemicals is an interesting category because, just because it's natural doesn't mean it's safe. You have to prove it. So when you have let's say a plant extract or a natural biochemical, you have to petition the EPA and go to the Biochemical Classification Committee, which deems whether it's suitable to be registered as a biochemical or a chemical pesticide instead. And what's the difference? Well, you have to show that it has a non-toxic mode of action to the pest or the pathogen in order to be registered as a biochemical. And so how do you have a non-toxic mode of action to the pest? Well, you could be a pheromone, a mating disruption chemical. That's a biopesticide, a biochemical. Or in our case of regalia, showing here knotweed on the slide, which is our product regalia is an extract of knotweed. It boosts the plant's immune system through induced systemic resistance or systemic acquired resistance, and therefore is not directly killing the pathogen. That's a non-toxic mode of action. Now, in the 70-year history of registration on use of biopesticides, there's actually never been an environmental or health incident. So these are well regulated. US EPA, fortunately, has the most efficient regulatory process, so we can get through quicker than most places. What's required? You have to prove it. You have to prove that these are low risk. We start at the exact same place as chemical pesticides, and you have to start with tier 1, toxin ecotox. And if there's any toxic effects in this tier 1, you have to do more studies, tier 2, tier 3, just like you would a chemical pesticide. But most small companies can't afford to do expensive toxicology studies, so are going to be developing products that have a non-toxic mode of action in this, or non-toxic, no direct toxic effects in this tier 1. So we have to do the rat studies, product chemistry, a 5-batch analysis. So you have to show the EPA that you can produce this consistently over 5 batches that generally are commercial. They could be semi-commercial or commercial batches. And unlike chemicals, which actually you do not have to show that there's no human pathogens, chemical pesticides, we have to prove that there's 0 human pathogens in our products. And we have to do, also unlike chemical pesticides, we have to do full 30-day feeding studies for all the ecological organisms, the ecotox studies, the birds and the fish, the daphnia, honeybees and lacewings, lady beetles and parasitic wasps. And believe me, it's sometimes a real challenge to do 30-day feeding studies of these organisms. We have to do an endangered species review, and you have to prove that your product can be exempt from tolerance and prove that it can be safe for food. The California Department of Pesticide Regulation is the one state that requires efficacy data, and all countries outside the US all do as well. Now, organic often causes confusion because there's a number of labels out there, but on the biopesticide bag or jug, you can have 2 types of labels. The first one is the National Organic Program label, which is for organic production. You also see can be used for organic production. That's the triple leaf logo. That is the one that is nationally regulated. And when you submit to the EPA, they will review for the USDA's National Organic Program whether it can be listed for organic. So then you'll see that triple leaf logo. Now, OMRI, or the Organic Materials Review Institute, which is a nonprofit that also lists products for organic. For biopesticides, if you have the triple leaf logo from the NOP, you don't need OMRI, but most growers recognize the OMRI more than they do the triple leaf logo, so most companies will get both of them. Now, if you're a biostimulant, you're not going through the EPA, so you wouldn't have that triple leaf logo, but you would also, or a biofertilizer, you would need that OMRI to show that you're organic. And then, of course, if the farmer is using inputs that are listed for organic under NOP or OMRI, then you can get the USDA organic seal, as you see here, for food, or the farmer can show that they are certified by a certifying agency, such as the California Certifying Agency, CCOF. Now, what's not registered as a biopesticide but are natural products? Well, there's a number of them. The product, a very effective product that is used by a lot of organic growers I know under the brand Entrust, now owned by Corteva, originally a Dow product. This is an organic formulation, and it is molecules produced by the microbe, Saccharopolyspora spinosa. But because it is got a toxic mode of action, works on the GABA nervous system of the pest, that is considered a toxic mode of action, therefore registered as a chemical pesticide, not as a biochemical biopesticide. Likewise, pyganic, or from the pyrethrins, from extracts from chrysanthemum flowers, also organic, can be used in organic. However, it is, again, toxic mode of action, so registered as a chemical pesticide. It works on the sodium channel of the insect. That's considered a toxic mode of action. Now, other examples here, abamectin, also toxic mode of action, fermentation-based product. Tebufenazide is a mimic of a juvenile hormone in insects, but because it's different than the natural and synthetically made, it's considered a chemical pesticide. And then azoxystrobin, the strobilurins, a fantastic group of fungicides that was originally derived from the compound from a mushroom, but then modified synthetically. That's considered a synthetic pesticide because it's not nature identical. So why should we look at bio anymore to look at for pesticides, biostimulants, natural products? Well, compared to pharmaceuticals, where more than 50% of your human drugs are derived from natural sources, like the heart drug from digitalis plant, foxglove, as you see here, or Taxol breast cancer drug from the Pacific Yew, as you see here, or antibiotics or penicillin, many of your drugs. Only 15% of pesticides are derived from natural sources. So we have a wide world out there. We're really at the very early stages of being able to utilize natural products for pest management. And because we have molecular tools and so many scientific tools today that can enable discovery and development, you can discover and develop these things much faster and more with more knowledge than we were able to in the past. Now, switching to what we do at Marrone Bio Innovations, I started the company in 2006. We now have quite a range of products and are growing quickly and expanding our sales. We're 90-10 US and international, and we're expanding rapidly outside the US. Biologicals are growing faster outside the US than in the US, although the US is still growing, but it's a more mature market. We've isolated about 18,000 microbes. Every company I've started, and actually even at Monsanto, I screened microorganisms, and so every company I've done, every time we start screening, we get better and better and can screen fewer microbes to find the products that we commercialize. And we have a fermentation facility, which we built in Bangor, Michigan. And we have, in September, we bought a Finnish company that has uses pulp and paper wood waste to create a macromolecule, that polymer that we put in small amounts of micronutrients and used as a seed treatment. So how do we do this? Well, we are kind of old-fashioned in this way. There's a number of companies that will use genomics up front from samples for their screening, but we actually think the old-fashioned way, which is bringing back samples to the laboratory, testing them first for biological activity, fermenting them, and then testing them against a range of target pests and pathogens. And then when we get activity, we do the genomics and do a lot more of the natural product chemistry and genomics. And we find that has worked really well for us, because just because something has a particular gene doesn't necessarily mean it's being expressed. So focusing on good biological activity up front, and then doing applying molecular tools after that to sequence our hits and candidates is the way we've been very successful at doing this. We have a natural product chemistry group that's absolutely critical to this, because every microbe or plant extract we discover, we want to know what the active molecules are there. And I'll talk about that in a minute. And then we do a lot of these functions in parallel. So we have teams of groups of scientists that are working in parallel to develop something. So while natural product chemistry would be working, we'll also have the fermentation process development folks learning how to scale the microbe, and then the formulation group, how to formulate it, regulatory working on the tox studies, and then as we get the first process, we'll pass it off to our manufacturing group to scale in a larger scale, although we have pilot scale here in Davis as well, up to 100 liters. Now, because this is the American ACS seminar, I really want to touch on how important natural product chemistry is here for us. A lot of companies are developing biologicals now and microbes, and a lot of them are focusing on living microbes without ever understanding what compounds are being made by the microbes that's causing pesticidal activity. Some microbes will colonize plants, so you want them to be alive, but most of the time they do produce compounds, so you want to know what's happening. So we feel that it's critical to having a consistent product, so that every batch we make, we know a specific level of the compounds for that efficacy. And so we develop analytical methods for the bioactive chemistry and use that in our quality control in manufacturing. And then the beauty of this is that we can continuously optimize the compound titers and their potency by our teams, and then have that continuous improvement of the product I talked about. So without gene editing or genetic modification or recombinant DNA, based on what we know about the gene sequence of the microbe, we can amp up those compound levels and those titers hundreds of fold, and have a much more efficient product. For example, we have a nematicide insecticide that we have more than 500-fold increased the compounds, and that takes the rate for the farmer from a seed treatment from 6 ounces down to 1/3 of an ounce, or on a foliar insecticide from 1 to 4 quarts down to ounces per acre. So this is the power of being able to understand the chemistry and then continuously optimize. So I'm not going to spend much detail about our products, but just to give you a flavor of the types of natural compounds and sources of these. We purchased some sanitizers. Now one of them is being used for sanitizing for actually approved by the EPA for coronavirus, jet oxide 15% peroxyacetic acid. And we purchased that from a small company last year. Our knotweed extract, regalia, and pace setter for row crops. We just launched the pace setter brand. It's a great product for improving crop health, controlling plant diseases. Bacillus nakamuri, the basis of Stargus, we discovered from a Northern California rice field. Chromobacterium subsugi, the new species, the basis of our Grain Devo bioinsecticide, was discovered by the USDA from under a hemlock tree in Maryland. Venerate, we discovered from a garden near the Rhinogen temple, hence Burkholderia rhinogensis, a new species of Burkholderia. This is a killed microorganism, so we're delivering the natural products without it being alive. And then our brands for nematode control, Magistine and Zelto. Haven is an extract of coconut oil. When sprayed on the crops at bloom, or when the crop, like corn, is rapidly growing, can, or shortly after bloom, like on almonds, reduces crop stress and dramatically improves yield. And I mentioned the ProFarm acquisition with our wood pulp products that are used as seed treatment at very low doses and low rates, and boosting crop yields quite well with this matrixed substances. And then Zequanox is the one product we have from that's not ag, and that is a Pseudomonas fluorescens strain that produces proteins and some other compounds that disrupt the feeding of the zebra and quagga mussel when they feed. Now, what do we actually patent? This is very important. It's a very competitive industry. You don't want someone isolating a microbe from your product and then selling it as their own, or finding another microbe with the same compounds. So we do patent the microorganisms. It's harder to patent them because there was a ruling by the Supreme Court on this that made it much harder, but we still can get patents on claims on microorganisms if you know how to do that. But even more important is getting the claims on the compounds. This gives very powerful intellectual property. So we do patent all the compounds, and they can be known compounds that have a new use. We can find a pharmaceutical compound that has a new use for pesticides, that's patentable, or a new use on a new pest or a new pathogen. The mixtures are patentable, and mixtures with other products, synergistic mixtures, is patentable, and formulations are patented. What we do not generally patent are the fermentation processes. These are kept as trade secret because they're so key to how to make the product work. We have teams in R&D that we call microbe-centered SWAT teams that are support and enhancement of the commercial products. As I mentioned, we'll have version 1.0, 2.0, 3.0. So the teams are constantly improving the products. They're understanding the genetics and microbial physiology of the microbe to be able to improve its yield and potency in those compounds. So we'll constantly be finding, characterizing the active chemistry and improving it. And that goes into gross margins, improves the profitability of the product, as well as I said, lowers the use rate. Understanding the mode of action, so the farmer can understand what is actually happening on the pest or the plant pathogen, and or on the plant. Many of times these products turn on genes in the plant, and so our molecular biology group, a plant pathology group, will want to know what's actually happening on the plant as well. These are very complicated multi-mode of action products, so we have teams constantly working on these together. Our one microbe from the Buddhist temple in Japan, Burkholderia rhinogensis, is remarkable in that it produces herbicidal compounds, insecticidal compounds, and nematicidal compounds. And we can shunt the fermentation, different processes, to shunt it to the different compounds. Obviously, you would not want a herbicide in an insecticide and nematicide, so we can find ways to reduce or eliminate the herbicidal compounds in the fermentation. But then when we want the herbicidal compounds, we change the fermentation to amp up those specific molecules. So the power of today's molecular biology and tools. Now, one of the breakout areas for biologicals is in the seed treatment category. And it's a very competitive space, with every major company wanting to have a biological stacked with a chemical on the seed. So here's an example of synergistic activity of microbes with chemical on the seed. Although we have developed, as has our partner Alba, under the BioST brand, also developed an all biological seed treatment that can be used by both organic and conventional growers. But the vast use of these seed treatments are stacked with chemicals right now. And so we have last year about 5 million acres with one of our insecticide nematicide microbes in the Alba BioST brand. So this has been a very successful area for us. Now, I point out that sometimes farmers get confused by the number of bacillus-based products that are on the market. There's a lot of them. It's much easier to discover a biofungicide than it is to discover a bioinsecticide. Discovering a bionematicide somewhere in between, but it's pretty easy to discover a fungicidal bacillus. So there's a lot of these out there, but they're not all the same, which is why the EPA and every regulatory agency in the world regulates the strain, not the species. That's the specific strain. So with the exception of the Bacillus subtilis GBO3 and the Bacillus amyloliquefaciens ENV503, which are identical and commercialized as two different products, two different product brands by two different companies, all of the rest of them are different and have different characteristics despite being similar bacilli. And here's an example of using chemistry to distinguish. So we have Stargus, our Bacillus nakamuri, strain F727, the one from the rice field, and its chemical profile, lipopeptides and other compounds, is distinctly different than two other products, which I call product A or product B, which were two of the products that are listed on the other slide. So you have to definitely look at the chemistry in order to distinguish your bacillus from others, because there's a lot of patents in this area, and you don't want to run afoul of commercializing something that might have the same chemistry as another product. So here's a way we can distinguish them. Now I'm going to talk about the unique modes of action of some of these types of products, and I'll use the Chromobacterium subsugi in Grandivo, the bacteria discovered from under the hemlock tree by the USDA. This is a very unique bacteria. It produces violacin, which is the compound over here on the left, and also the analogs of this. And it is repellent to the bugs. They hate it. So we had to develop a special bioassay. Here's a repellency assay using aphids that we developed, because just using a typical knockdown kill assay wouldn't work here. So we had to, and we actually use this in quality control, use this repellency assay. It works very well. It's dose dependent. Now on the right side, this big monster molecule, this chromamide, which is novel, we patented that, is causing on this leaf beetle gut disruption and also kind of diarrhea over here. And so the combination of these chromamides with some proteins that we've discovered, one of which is novel, synergize each other. So the chromamide alone is only about 40% effective against the insects, but you add these proteins to it and it boosts it up to 100%. So all of these molecules work together in a cocktail. It's very complex, which is why our natural product chemistry group is constantly working on this arena. It's just not one molecule doing the work. So here's showing you how it affects the reproduction of aphids compared to another natural product, but a chemical abamectin. And so there's a dramatic reduction in the number of aphids. But we needed a unique bioassay to be able to test efficacy, since we couldn't use just a regular kill method. This product does not kill within 24, 48, 72 hours. Spotted wing Drosophila, this showing you how there's a dramatic suppression, two different formulations of Grandivo on egg laying of the adults. So this is, again, if you're just looking at a knockdown of the flies, you're not going to see it. But if you look at the suppression of the whole generations of the flies, you'll see great results. USDA did some great work on putting Grandivo on a glass slide and then having the brown marmorated stink bugs walk on the slides, and compared to other treatments and water, dramatic movement. And so they showed how dramatically these bugs hated this product and tried to get it off of them, but their preening behavior, they were able to, this only works by ingestion, they were able to get enough product on them, in them, to be able to start killing them. But it's slow kill. So you don't see that full kill until the 7 day point and 7 to 10 days. So which is what we advise when we talk to growers or cooperative extension or pest control advisors about how to use this product. But so the product will provide a wonderful season long effect on the total reproduction and the suppression of the whole population. But if you're just looking at it like it's going to kill something in a quick knockdown, it's not going to show that. So apply it early when the adults are out there starting to lay eggs versus trying to knock it down when there's already a high population. So this means that the traditional integrated pest management rules or thresholds might not apply. So often the practitioners will say, build up your pest to an economic injury level, and only then do you come in with a pesticide. Well, some of these products you have to start a bit earlier and work on a more preventative manner. Another consideration is how much water to use. Because you're trying to get a lethal dose on the leaf for the bug to ingest, more water is not better, as it shows in this slide. 50 gallons per acre outperformed in terms of marketable fruit, strawberry, against mites versus the higher levels of water. And so more is not better. You want to deliver that lethal dose. And then which adjuvant to use? Farmers throw in often adjuvants to make the product stick on the leaf, be more rain fast or whatever. And we found that there's a dramatic change in the LC50 depending on which adjuvants used. So it's something else our R&D has to do when we're developing a product. Also, dramatic effect on formulation. Because these are natural compounds, some of the compounds in Grandivo are gone within hours. So we have to put a sunscreen in the product to prevent it to have it last long enough on the leaf to do its thing. And here's a dramatic effect of the different formulations and the number of worms. And if Coragen is one of the very best insecticides on the market, and our formulation number 4, which is our current product, is not statistically different on armyworms and loopers than this product. So we're able to dramatically change the efficacy on getting a better formulation. And again, that's in our version 1.0, 2.0, 3.0 process, how we can do that. Now, here's an example. I remember at the early part of this webinar, I mentioned that these products perform in integrated programs. They excel in programs together with other products and conventional products. Here's a perfect example of that. Now, we want to know what our product does on its own, but that's not the end of the story. Unfortunately, most of the time, these products are done standalone against the best cocktail of chemicals, and then that's it. But that's not the way farmers use products. Farmers use products in integrated programs. So what we've done is added Venerate, our other novel bioinsecticide, to the traditional program and looked at the results against navel orangeworm, the biggest problem on almonds today, and mites. And so what we showed was that yes, our product works alone, but we can boost the performance of the traditional program by adding Venerate to the program for navel orangeworm or mites. And we called that BioUnite because one would think that this is because farmers mix and match chemical products all the time. What we found is that it was actually a paradigm shift with the key influencers and pest control advisors and cooperative extension and growers to mix and match biologicals with chemicals because again, these products are typically pigeonholed just for organic. So we did have to do a lot of education and paradigm changing to show the benefits of these products integrated in. Just what I said, many existing IPM programs are outdated. They don't reflect the products and the modes of action available to today's growers. Trials and demos should be done integrated and rather than just standalone comparisons. Don't get me wrong, you want to know what your product does alone, but these products shine in integrated programs. Now to wrap up, I'm going to move into a big area that's a hot topic today. You're going to hear all kinds of healthy soil initiatives. The California government has a healthy soil initiative. There's global initiatives. USDA has announced some innovations on soil health, but there's a lot of science behind soil health that we don't know, but we're learning. So there's companies, startup companies, 3 here, Trace Genomics, the oldest one. Then there's Biomakers, and then a new one from Taiwan, Yinong, that are analyzing the DNA, the microbes in your soil, and they could have a farmer actually send in a soil sample and find out what's going on, and then kind of give a prescription or find out what the biodiversity is, how poor the soil is based on the biodiversity of the microbes in the soil. So this is at the early stages of how growers can use this to enhance and understand what's going on in their fields. Now, a very interesting example from our company, our molecular biology group did a great study with Muscador albus, which is a fungus discovered by Dr. Gary Strobel at Montana State University, emeritus now. And we have a strain SA13, which this work was done. And it produces a number of volatile compounds. You could put this fungus in the soil. It fumigates the soil, kills nematodes, and as well as a lot of plant pathogens. And we've done a lot of trials to show it's a great biofumigant. But what our group did was that we've got the immediate question is, well, what are you doing to all the microbes in the soil and the biodiversity in the soil? So we have actually we had a student intern that worked with us at from UC Davis who did the full gene sequence of this and also helped do this study, and showing that pre and post fumigation of putting this in the soil, on the right-hand side, we're showing the dramatic drop in plant pathogenic species. Notice the orange one is Fusarium, which hangs around a little longer but eventually dies out. All the rest of the plant pathogens immediately are reduced by this biofumigant in the soil very quickly. But on the left, that is showing you all the different beneficial soil microorganisms. On the very left is the untreated, and then all the way showing you what happens after fumigation. And amazingly, we can kill off the pathogens, but keep the beneficial soil microorganisms. Isn't nature grand? I love it. Another example of what's going on under the soil that's very exciting are the strigolactones. There's a company, Sound Agriculture, that is exploiting the strigolactones to make biostimulants. These are signaling molecules pumped out by plant roots that say, hey, mycorrhizae, I've got cake, I've got something really good for you if you attach to my root. And the mycorrhizae, which are beneficial microbes, fungus that enhances the crop growth and helps nutrients taken up into the plant better, come to the plant via these strigolactones. The strigolactones also attract witchweed, striga, hence named. So they're, now that we know these signaling molecules, we can design ways to prevent the witchweed and the striga weed, parasitic weed, from coming to the root. But at the same time, get the mycorrhizae to come. Indeed, the USDA, funded by the United Soybean Board, showed they can now breed for higher levels of these compounds in a crop like soybeans. So on the left, they've bred in the ability to have more of these signaling molecules that attract the mycorrhizae, and on the right, not. This is very exciting stuff. So I gave a talk not long ago to a seed production course at UC Davis. And my message was, when you're breeding now, you better be looking at the microbe interactions and these signaling compounds. This is a big part of the story now. Another example, which I love, from Dr. Hofta at Ghent University, showing how she's worked for a long time on cyclic lipopeptides from Pseudomonas fluorescens and other Pseudomonas. And she's showing you that higher quality soils have more diverse chemistries of these cyclic lipopeptides, and different cocoyam varieties have different fluorescent microbes, Pseudomonas, associated with them, and that the white cocoyam really succumbed to Pythium root rot, but the red didn't. And why? Because it supported a more diverse cyclic lipopeptide spectrum. Very exciting, again, that you're able to now tease out when you're seeing why some varieties of crops are doing better than others. Another example from Linda Tomashow's lab up at USDA and Washington State, showing again with Pseudomonas fluorescens that the suppressive properties of soils that over time were suppressing take-all disease of wheat. So she's teased out and her teams have teased out the compounds causing this and showing you the phenazine compounds that are responsible for the suppressive activity. And again, certain wheat varieties are better at others from attracting these bacteria. Same with fusarium. Fusarium wilt suppressive soils had a lot more of these compounds. So biopesticides today, very exciting. Hope I've convinced you that this is a wonderful area to be in today and the value of these biopesticides for crop production. But there's a lot of challenges, and I've also mentioned a lot of challenges in developing them. And so, but we have a lot of opportunity now with what we know and the constantly changing science and improvement of molecular and tools, formulation tools, material science, etc., in being able to drive improvements through research and development and creating products that can drive costs down, can compete with chemicals, can be lower rates per acre, and improve yields and qualities. And it's not just biology and chemistry, though, because there's a lot going on in precision farming and big data. And we're just at the very early stages of integrating precision tools and big data to integrated pest management. So over time, we're going to be able to better predict pest populations and when they come in. We can see them with remote sensing and vision systems. There's companies like Semios that has a vision-enabled trap for moths that pest moths, and then they can spray when there's a high population and do it all from the computer, and the farmer doesn't even have to walk the field anymore, which has its own implications, I guess. And analyzing, using big data, analyzing what's going on in the soil. And then a lot going on are in spraying technology. Our ability to spray pesticides has been very crude and at where to get target pesticides at where they need to go, but there's a lot going on in smart sprayers. This is a very exciting area. So the world of ag, of tech and biotech, and biology, chemistry, and precision tools are all merging together in exciting opportunity to solve problems on the farm. A lot of companies working in the biological, biochemical area. Here's just a few of the young companies working on microbials for soil and seed. So today's biologicals address all key issues in food production and aligns very rapidly with the environmental, social, and governance investment category of investors in really scrutinizing companies and how they treat their workers, how their products affect the environment, what are they doing to carbon footprint, and so on. So it's exciting to be in an area that meets right at the cutting edge of what's going on in the needs for more sustainable food production. To get more information, Biologicals Product Industry Alliance, BPIA.org, is a trade group of biopesticides and biostimulant companies, and that I started in the year 2000. Now quite a big group. And then our European counterpart is IBMA, and they're both trade groups that are dedicated to for advocacy and of both biostimulants and biopesticides. So if you would like to contact me, all my contact information is here. I'm happy to provide a PDF of my slides, but if you want, you can read. Steve edited this for me, Steve Duke, and it is a very comprehensive overview in pest management science, which was published last year, Pesticidal Natural Product Status and Future Potential. So if you would like further reading. Thank you very much.